Hi everyone. Today will be today I'll be showing you a demo on constructing a pangenome graph using Minigraph cactus as a tool, and we'll be constructing the graph for the C4 genes. So what are C4 genes? These are the genes that are present in human chromosome 6, and they are responsible for the immune response of an individual. And this particular region is highly polymorphic, and because of that, this particular region is kind of very different in every individual. And that is why this particular region is of interest to the biologist. So, we will be comparing our output with the output that was generated in this paper on the draft of human pangenome reference, and I will show you the output on bandage, and I will also show you the path for this haplotype GRCH38 in the output. The dataset that we will be using is from this particular record in Zenodo, and the tools that we will be using are AGC and Minigraph cactus. So, let us begin. So, first of all you have to install these tools AGC and Minigraph cactus. So, you can go to their GitHub repository and then install it. I will also provide the shell script to install the Minigraph cactus in case you are not able to install it directly from the repository. So, I will quickly walk you through each and every command, what is happening in this command, but before that I will run this script so that we can get the output in that time. So, I am actually running it over a remote server, and the job scheduler there is PBS. So, we will be doing this. So, first of all we will be using AGC as a tool, and then extracting the file in the .agc format and getting the FASTA files out of it in this particular path. After we extract the FASTA files, so we have to construct the this overall consensus of these sequences using the tool Minigraph cactus. So, this particular tool takes the sequence file as an input. What this sequence file is? It comprises of the rows, where each row represent a sequence and its corresponding FASTA file path in your basically folder, and these two values have to be separated by a tab character. For example, I have created one before this particular demo, and it looks like this. And then it will also ask for the reference along which we will have to align all the other sequences. So, for this particular experiment I am using C4 CHM13 particular haplotype as a reference, and I have also passed the path where this GFA file and all other files that this tool will print will be stored. So, it is running, and basically it takes like 3-4 minutes to actually create the graph and all. And in the meantime I will start with the tool bandage. One second. So, yeah. So, I will quickly walk you through the GitHub repository of these tools by the time this stuff is getting executed. So, once you go to this repository, you can directly go to this binary, the location where the binary is located, and you can download the binary corresponding to your architecture and save the binary file in your bin folder, and using that you will be easily able to access that binary from any location. The same is for Minigraph cactus. Over here you need to go to this particular location, and I am using this particular legacy binary. You can use this other things as well as per your architecture. And over here the steps are like it involves some more steps to install the cactus tool. So, after you get the binary, you have to also create a virtual environment, and in that particular virtual environment you have to install other requirements, and this is mentioned on the GitHub website. And okay, so let us check if it has, okay, it's still running, but yeah, so let it run. I have already generated the output, and so the c4.gfa file. So, the output will be in this particular format. So, if you so each file has these particular parameters, where S represents that particular segment and the nucleotide sequence corresponding to that segment. L is the link, that means the edge bit, it will mention the between which pair of segments there is an edge, and then there are information about the path and the walks. So, using this particular link, it will create the graph considering the segments to be nodes. So, if you see here, S lines are there in the start, followed by, if you go down you will find L lines. So, these are the walks, and then in the end these are the connections, basically the edges between the nodes. And we can open this. So, Bandage is a tool to plot this sort of use a GFA file as input and plot the graph corresponding to that. So, let me load it quickly. One second. Wait, not here. So, you go to your folder wherever you have stored that file, and then you load it. So, you use the command draw graph to draw that particular graph, and it will create the graph. So, let me reduce the size. So, if you see here, right, the output is kind of similar to the output by Minigraph cactus. So, it has two long tails and followed by these loops, and that is what we are observing. It is not exactly the same, but it is kind of similar. Now, in order to highlight a path corresponding to one single haplotype, one easy way is that you have, there are multiple other options, but one easy way here is you have these walks corresponding to different haplotypes, right. So, what you do is you copy this thing, copy the walk, and since these are like there are brackets between the two values, you have to replace that bracket with a comma. So, you can write a Python script to convert that particular string to a comma separated string, and you will get an output like this, right. After you get this output, you just copy paste this output in the find location of the nodes, this particular text box, and you find these nodes there, and then you can express like set color of these nodes to be different as compared to the other nodes in the graph. And if you, one second, if you check, then so this is the path that is actually representing the haplotype GRCH38, and if you check here as well, so it is kind of similar, but not exactly the same. So basically it actually covers both the tails, and that is actually represented here as well. So this was a short demo on basically using tools Minigraph, cactus, bandage. So one important point here is that bandage won't work with bigger graphs. So that's why we chose C4 genes as an input. And yeah, so thank you everyone.